Welcome to the stage, our first overgraduate finalist, Estonia-TUIT.
So, hello everyone. Honestly, we can't even believe that we are standing on this stage, guys. We are iGEM team Estonia-TUIT, and we are buzzing with excitement to share our BeeYeast project, yeast engineering to fight bee viral infection. You know, bees are not just famous for their sweet honey, beeswax, propolis, and bee venom. They are also the original economic bees, responsible for billions of dollars in food production. Can you imagine that the impact of the bees on the economy is estimated to range from 235 to 577 billion US dollars annually?
Wow, that is actually unbelievable.
Yeah, but here is the catch. The bee population is declining faster than you can say honeycomb. Blame climate change, excessive use of pesticides and pathogens. One of the most widespread viral pathogen is a deformed virus that infects the bees and causes the death of millions of bees population every year. This issue is worsened by external parasites known as varroa mites, which infect larvae, pupae, and even adult bees, transmitting DWV and weakening their immune system. The combination of mites and DWV possesses a significant threat to pollinator populations and agriculture.
Those mites should be like the nightmare of the bees. They're almost like Freddy Krueger in their world.
Yeah, exactly. And it's a real bee pandemic, and we need to do something with it.
So it seems that there is a need for an innovative approach to safeguard bee populations.
Yeah, well, it all started with synthetic biology. We decided to use it to help bees fight viral infection. We looked into RNA interference, one of the bees' innate antiviral response, where small RNA fragments bind to messenger RNA and shut down viral translation. Upon viral infection, the viral genome is replicated in the bee cell, leading to the production of double-stranded RNA. In B cells, the dicer binds to the double-stranded RNA and produces small interfering RNAs. However, these shRNAs are produced only after affecting the B cells, which makes innate RNAi machinery insufficient. That's why our team aimed to enhance the natural antiviral response. And this is how we set out to do it. We modified these cells to produce a shRNA against the DWV to supply B cells with them before the infection. We started by screening the deformed virus genome to select one of the best candidate sequences which can be used by the RNA interference machinery. Next, we cloned this sequence into our yeast plasmid vector to express a short hairpin RNAs in yeast. Then, we introduced the gene encoded for dicer protein to cleave the hairpin from shRNA and produce active siRNAs. To deliver the synthesized siRNAs to bees, we plan to feed them with pills made out of our genetically engineered yeast or inject siRNA-containing extracts directly to honeycombs. When delivered to cells, our siRNAs are then processed by Argonaut, one of the proteins from the RNA-induced silencing complex. Argonaut stays associated with one siRNA strand and then guides this complex to the viral mRNA, binds to it, and resulting in its cleavage and suppression of the infection. Additional supplementation of antiviral siRNAs should promote robust RNAi response as soon as the virus enters the cell, providing better protection from the infection. So, modeling is like the GPS for our project. It helps us navigate the RNA interference highway. To strategically plan and optimize our project, we employed modeling techniques for computational simulation of siRNA production in yeast, along with the key steps that happen in B cells. This allowed us to determine the initial experimental setup and evaluate the performance of the proposed approach. Moreover, modeling shRNA production in yeast allows us to optimize various steps of the project and processes, including the choice of promoters for shRNA and dicer, potential concentration, efficiency of shRNA folding, and its stability. We divided our project into 4 main stages and enabled to pay attention to every of the steps. Firstly, modeling of shRNA production revealed the considerable difference between the 10 designed shRNAs and showed that the folding process is not a bottleneck in shRNA accumulation, as the concentration of unfolded shRNA is over 3 orders of magnitude lower than the concentration of the folded shRNA throughout the process, which means that almost all of the shRNA was folded. It's success. During the subsequent phase, it was predicted the dicer expression controlled by TEF promoter is sufficient to cleave most of the synthesized shRNA, converted it to shRNA. Thirdly, previous studies have found that delivering synthetic shRNAs to bees reduces DWB-related mortality, indicating that the small RNAs are capable of passing through the digestive system with retaining activity. By modeling shRNA uptake by B cells, we found that the yeast-produced shRNA is expected to accumulate in the low nanomolar range. Importantly, that has been shown to be sufficient for effective RNA interference, whereas high concentration are more prone to cause off-target effects. And finally, the modeling of RNA interference in bees showed a strong dependence of antiviral response on shRNA concentration, suggested that supplementing shRNA is likely to have a strong beneficial effect on bee antiviral defense. We also modeled the same mechanism on our test system, Yeast, to make a correlation between our wet lab and dry lab, and conducted the antiviral response in bees. Taken together, our modeling efforts guide us on which promoters and expression levels should be used in our engineering yeast, how to set up our experiments, and inform us on the shRNA concentration we need to aim for.
So, testing the effectiveness of our designed shRNAs in suppressing deformed wing virus in B cells would be labor-intensive, costly, as well as a complicated process, mainly due to the involvement of working in live virus cultures. Therefore, we designed a reporter system in yeast, Saccharomyces cerevisiae, to assess the impact of these shRNAs. Since Saccharomyces cerevisiae do not possess their own RNAi machinery, in the initial phase, we introduced dicer and argonaute proteins, which are essential elements for RNA interference. This resulted in the engineered yeast strain capable of enzymatically cleaving shRNA and converting it to siRNA molecules. At the next step of our project, we created a plasmid containing an shRNA sequence under the control of the galactose promoter. This plasmid is responsible for expressing specific shRNA molecules that are capable to target particular regions of the deformed wing virus RNA. To facilitate a quantitative readout of the efficiency of siRNAs against deformed wing virus, we created the third component of our system, a reporter consisting out of the gene coding for green fluorescent protein fused to the viral target sequence in its 3' untranslated region, right after the GFP stop codon. The reporter expression, as well as shRNA, is controlled by the inducible pGal1 promoter. By utilizing this system, we aim to assess the degree of inhibition exhibited by the siRNAs against complementarily viral RNA derived from the DWV genome. The system can be used to search for the best siRNA candidates against the target, and potentially can be expanded for the screening of efficient target sequences for other viral pathogens. According to one of our amazing computational models, the concentration of siRNA is expected to reach highest levels around the 24-hour mark. Therefore, we measured the GFP fluorescence in yeast cultures 1 day after either inducing or repressing the galactose promoter. Growing cells in glucose leads to low background level of GFP fluorescence, proving that the galactose promoter is repressed. When the expression of GFP reporter and siRNA is induced by galactose, the intensity of GFP signal depends on the efficiency of our RNA interference. If a particular small interfering RNA is efficient at binding to its target sequence, the argonaute cleaves the GFP mRNA, leading to decreased fluorescent levels. On the other hand, if the siRNA is not that efficient in finding the target sequence, or we deliberately mismatch them, the GFP mRNA is translated, leading to high fluorescent signal. We observed that strain lacking the RNA machinery proteins but capable of producing the GFP target and shRNA showed elevated levels of fluorescence. Also, in the strain lacking the short hairpin RNA plasmid but containing dicer, argonaut, and the GFP target sequence, the fluorescent levels were high, just as we expected.
Truth to be told, our experiments is like a roller coaster of emotions. We tested 7 siRNAs, and guess what?
Drum roll, please.
4 of them shows the sufficient suppression by the lowers the level of GFP. That state that suggesting at each partial suppression by our designed siRNAs.
So, the results of our experiments led to 3 important conclusions. First, the presented work validates the published results that introducing dicer and argonaut proteins to Saccharomyces cerevisiae is sufficient to constitute an RNAi response. The experiments showed also that viral sequences introduced to the 3' untranslated region of a GFP coding transcript can be targeted by our siRNAs in yeast, making this a simple way to test the efficiency of different variants of the said siRNAs. We observed partial suppression on GFP reporter expression with tested anti-DWV siRNAs, confirming that they are strong candidates for further studies.
So, what's our next agenda?
Well, we are aiming to make our yeast-based siRNA testing system even more efficient by placing the GFP target and shRNA transcriptional units under the control of differing promoters.
We will also be testing our siRNAs based on the full length of DWV genome, and also investigating how our bee respond to these little RNA barriers.
And of course, we'll be watching out for any unexpected bee side effects. We don't want any bee drama. Safety is also our top priority. We keep everything at biosafety level 1. Better safe than sorry. But what about real-world needs?
Well, to ensure that our project aligns with responsible practices and real-world needs, our team has undertook several key human practices activities to shape our project. Firstly, we explored the apiculture regulation in Estonia, and we also consulted with experts from Agriculture and Food Board. Then we reached out to 10 beekeepers from 4 different countries to gather the insight of treating the viral diseases and assess the interest of stakeholders in our product. Their approach, their feedback, and our findings have shown that the deforming virus is really widespread across Europe and highlighted the need in novel treatment of infectious diseases. Encouragingly, some beekeepers have demonstrated the keen interest in our project once it's proven to be safe. We also partnered with experts from different fields such as virology, bee biology, RNA interference research, and mathematics, and incorporated their approach in different parts of our studies such as modeling, design, education, and safety. To fill the gap between science and society, our team organized, designed, and participated in already traditional public events, educational fairs, and info sessions. We conducted workshops, organized art exhibition, and escape room, and also we are proud to say that this year we also hosted the summer school. We aim to find the new approaches and education that will not only be academically informative, but also easily accessible and entertaining for everyone. For that, we have developed our own new educational materials, board games and online games. We have translated them into different languages, 10 different languages. We have used them in our public events, and we also made them available on our wiki. While designing those educational materials, we followed our team's teaching philosophy statement and aimed to include more people into science-based approach. For example, our workshops were designed that way that they were not limited to only a few senses, but encourage participants to interact with the subject that they are studying on the multiple sense levels, such as eyesight, hearing, touch, and smell. Overall, we believe that bridging gap between science and people is a key to more prosperous society. Therefore, we are making world a better place.
During the design and implementation of our iGEM project, we collaborated with key stakeholders and specialists, integrating their insights throughout the various phases of our project. We also integrated the results of our beautiful computational modeling into the design of our experiments. Engineered yeast to produce efficient shRNAs aimed to block the expression of deformed wing virus genes in bees. Developed a functional reporter system in yeast that can be used to test shRNA efficiency. Designed educational materials, hosted international summer school, and conducted multiple public engagement events. Developed multidisciplinary hard and soft skills and expanded our network. And just as importantly, we shared many emotional moments and became friends for life. We want to thank everyone who contributed to the project. Supervisors, team members, external experts, sponsors, and of course our science and technology family. The project would not be possible without the team and all the people who helped us during the season. So thank you to everyone who joined us in this epic bee-saving quest. And remember, everything is impossible until it's done.
Thank you so much.
Thank you so much.